This is the second half of chapter 12. It's actually going to be the third video, so there'll be the second half of the chapter is going to be divided into two separate videos. And this part of the chapter focuses on exceptions to traditional Mendelian inheritance, which is what the focus of the first part of the chapter was. We worked through Punnett squares, talked about monohybrid and dihybrid crosses. So there are pretty common exceptions to those rules that we learned through Mendelian inheritance. So I'm going to give you some specific examples of those. At the very end of the chapter, it'll talk just a little bit about how prokaryotes, bacteria, for example, that do not sexually reproduce, how they can still transmit genes to each other through a quote-unquote mating process. All right, so the first thing I want you to consider is alleles. And so remember, alleles are different versions of a gene, right? You have the dominant allele of the A gene, right? The capital A version of that gene, or you could have the recessive version of that gene, the little a. And so in all the examples that we've looked at so far, you always have basically two different alleles for every gene, either the dominant, the uppercase, or the recessive, the lowercase. But, this is the big but, there are lots of examples out there in biology where a gene may have more than two alleles. And so in that case, how does that work? What does that look like? If we think of this just in terms of, well, how do we even get alleles? Where do these things come from? And they basically come through this process of mutation, which is changes to the DNA, right? So you can have, one quote-unquote original allele of a particular gene. Through mutation, you now get a new version of that gene that would be considered a second allele. But we don't necessarily have to stop there, right? We could acquire more mutations and end up with a third or even a fourth version of a particular gene. And as I said, there are examples of that out in the biological world. The term that geneticists typically use to refer to the allele that you tend to find most often in a given population is they'll refer to it as wild type. So we're going to talk about fruit flies later in this chapter. And typically fruit flies tend to have bright red eyes, so we would call that the wild type trait or the wild type allele in fruit flies. And then everything else, sometimes you'll get flies that have white eyes. We would consider those mutants. So, we don't tend to use these terms when we're talking about the human population because people get offended if you're calling them mutants, but, really wild type in humans is going to be brown skin, dark hair, brown eyes. Everything else is going to be mutations. So here's a kind of a classic example of multiple alleles. So this is referring specifically to the gene that controls fur color or coat color in rabbits, and you see something similar in a number of different other mammals. But in this particular instance, instead of just having two versions of the gene, we're using letter C for this gene, uppercase C, lowercase c, you actually have four alleles. So that gives us a bit of a nomenclature problem, right? Okay, well, if we usually use uppercase, lowercase to distinguish our two alleles, what do we do if we have more than two? And it varies a little bit field to field, but usually the accepted practice is, okay, we'll start adding these little superscripts. So as you can see from the way this is written out, you really have one allele that's dominant to all the others, and then you have three sort of recessive alleles that are different from each other. And every rabbit has two copies of this C gene. So depending on which two alleles it gets, you get one of these varied fur colors. And I'm not going to test you over this. I'm not worried about you memorizing exactly how fur color is determined in rabbits, but the way it plays out, if you're interested, is if you have one of the dominant alleles, a big C, you're going to be a dark gray rabbit. And then all the other fur colors we see here, you've got a combination of two different recessives. If you have sort of the traditional two lowercase c recessive, you're an albino rabbit, etc. So yeah, it contributes to that diversity, but of course it does make the genetics, if you're trying to do Punnett squares and figure out the inheritance patterns, it then makes things a little bit more complicated. But it's quite common to have this phenomenon of multiple alleles, we would call it. Another common exception to Mendelian rules is what's called incomplete dominance. And we've been, in all the examples we talked about earlier in the chapter, one allele was always completely dominant over the other, right? For example, if you crossed a purple flowered pea plant, that was the dominant trait, with a white flowered pea plant, you got big P, little p heterozygotes, and they were always purple, right? As long as you had one dominant allele, you always had the purple dominant phenotype. But that's not always the case. In this example with eggplants, you have a purple plant that produces purple eggplants. We'll call that dominant, big P, big P. White plant that produces white eggplants, little p, you cross those, and you get heterozygotes. But now instead of looking like the purple parent, the quote-unquote dominant parent, they have sort of an intermediate phenotype, right? It's sort of light purple or pink almost, right? We've combined the purple and the white together. So anytime you see this kind of thing where the heterozygote genotype gives you this intermediate appearance between the two homozygous parents, that would be considered incomplete dominance. Oops, yeah, there we go. Another exception is what's called codominance, and in this case, instead of the heterozygote giving you a blend of the two parents, in codominance, you basically see both of the parental phenotypes still present. So if we wanted to stick with like the color analogy, this would be as if you crossed a purple flowered plant with a white flowered plant, and the heterozygous big P, little p have like some purple flowers and some white flowers, or flowers that have purple and white splotches on them or something like that, right? You're seeing both of the parental phenotypes. One is not at all dominant over the other. It's not a blending. They're literally equals. They're co-dominant to each other, you would say. The example you see here is actually an example of multiple alleles and codominance in one, and this is the human blood typing. ABO blood typing is one of the very common ways to type human blood, which is, of course, is medically important, because if you have to get a blood transfusion, if you receive the wrong blood type, your body will mount an immune response against that, which could cause serious problems or even death. And so the gene we're talking about here in this instance is the I gene, and that gene encodes for these carbohydrates that get produced on the surface of red blood cells. And so I mentioned this was multiple alleles. The three alleles that you can see up here at the top. You have the dominant IA version, which produces the A carbohydrate, little triangles on the surface of the red blood cells. You have the dominant IB version, which produces a different carbohydrate, the little circles on the surface of red blood cells. And then you have a recessive I allele that is essentially a broken version of that gene, in that it doesn't actually code for any carbohydrate at all. So if you are homozygous recessive, little I, your red blood cells have no carbohydrates on the surface. Here's the codominance. If you are IA, IB, you produce both carbohydrates. So you're seeing both of those phenotypes present. You would be blood type AB versus O for the homozygous recessive. And then if you have either two of the IB alleles, or one IB and one recessive, basically all you're producing are the B carbohydrates. So you'd be type B, and similar for type A. Okay, so three alleles, multiple alleles, and codominance in that if you have the A and the B alleles, you see both phenotypes. Okay, so multiple alleles, codominance versus incomplete dominance. And we're used to thinking of this idea that each gene, is controlling one particular phenotype or one particular trait. And a lot of times that's true, but not always. There are examples of genes that are considered to have pleiotropic effects, which is just a fancy way of saying this one gene actually affects multiple phenotypic traits. And so I don't really know the biology behind how this example works out, but it has been demonstrated that the gene that controls fur color patterning in Siamese cats is also the same gene that is linked to the fact that Siamese cats often have crossed eyes. So they're both of those phenotypes are coming from that one gene. They're the same, that one protein encoded by that gene, so we'd say that gene has pleiotropic effects. The flip side of that coin is what's considered epistasis. And epistasis is where a single trait, a single phenotype, is controlled by two or potentially even more genes. Usually when you say epistasis, you're talking about two genes. So the example I'll give you here is fur color in Labrador Retriever dogs. There's two genes that control fur color, the B gene and the E gene. Okay. So the B allele or the B gene is producing a pigment protein. If you have a dominant version of the B gene, you make a black pigment. If you have recessive, you make a brown pigment. The second gene that's involved, the E gene, it produces a protein that actually deposits the pigment into the hair follicles. So you have to consider both of the genes, because if you are recessive for the E gene, which is the case down here with the yellow lab, you basically don't make a functional protein that will deposit pigment into the hair follicles. So it doesn't matter what's going on with the B gene, right? We don't know. This yellow lab could be dominant, right? Its cells might be making a lot of black pigment. That pigment just doesn't get deposited, so we end up with a white or a yellow dog, right? Whereas if you have at least one dominant version of the E gene, which is what you see in A and B here, you do make that protein that deposits the pigment. So then if we think about the color of the dog, now we have to look and say, okay, well, what's going on with the B gene then? What kind of pigment is it making? So if you have a dominant E, but you're homozygous recessive for B, meaning you're just making brown pigment, you end up with a brown lab, chocolate lab. And if you have a dominant E, again, you're making that pigment deposition protein, and you have at least one dominant B allele, that means you're making some black pigment, and you're going to be a black lab. So those two genes working together in concert to determine that one phenotype is epistasis, whereas one gene controlling multiple phenotypes would be pleiotropy or pleiotropic effect. And we're not going to get into doing Punnett squares with these kind of exceptions in this class, but if you go on to take genetics, at some point you will work on that. But it obviously complicates the Punnett squares and the analysis a little bit. Another big exception is environmental effect, right? So not everything is genetics all the time. Environment can be a very influential factor, you know. And when I say environment, could be light, temperature, nutrition, you know, you name it, right? You might have the genetics to be 7 feet tall, but if you're not getting proper nutrition, you know, as a child while you're growing, you're not going to have that phenotype that your genetics say you should have. Going back to Siamese cats, and this applies to certain breeds of rabbits as well, where you get this kind of point-restricted phenotype, meaning the extremities, right? The ears, the nose, the tail, and the feet are a darker fur color than the rest of the body. You get this due to an environmental effect where the enzyme that produces the dark fur is basically inactivated at higher temperatures, which would be the core of the body, right? You tend to be warmer there, so that enzyme gets turned off, basically denatures. And so you end up with white fur on the warmer parts of the body. When we were going through Mendelian inheritance, all the characters, all the traits we were talking about there were said to be discrete and qualitative. And what I mean by that is they always varied in just an either-or manner, right? So either the pea plants had purple flowers or they had white flowers. They had, green pea pods or they had yellow pea pods, etc. There was just those two possibilities. And as I'm sure you're aware, right, that's not always the case in the natural world. I mean, even in the human population, look at things like height or weight or skin color. Those things vary over a huge range, right? There's tons of different phenotypes, so we would call those quantitative characters. And anytime you see that kind of quantitative variation, this example is skin color here in humans, that almost always means that you have multiple genes contributing to that one particular phenotype. And in this example, in human skin color, we think there are at least 3 different genes that contribute. And basically the way this works out is the more dominant alleles you have of those 3 genes, A, B, and C, the darker your skin is going to be. So if you were completely homozygous dominant for A, B, and C, you would have very dark black skin. And on the flip side, if you were completely recessive for A, B, and C, you're going to have very pale white skin. But of course, there's lots of variation in between, right? Depending on how many dominant, how many recessive alleles you have. So that's quantitative variation, and that's very common. All right, so in the end of this first part, I wanted to shift our focus over to talking about fruit flies of all things, Drosophila melanogaster. And these have actually been a very, very powerful model system for doing genetics research. And a lot of the use of fruit flies was pioneered by this very famous biologist Thomas Hunt Morgan, who worked at Columbia University in the early 1900s. His research group was basically a Nobel Prize factory. So he got the Nobel Prize. Several of his students got the Nobel Prize for some of the work that they did with fruit flies. And I talked about model systems in an earlier chapter, but basically model systems are organisms that we may not be all that interested in terms of like medical application or commercial application, but they're organisms that researchers work with because they're easy to work with. They're small, they're cheap to grow and relatively easy to handle. And so with fruit flies, with Drosophila, they're great for genetics research because they interbreed like crazy and their life cycle is very short, right? So if you're doing genetics, you want to be able to look at, okay, what happens if I cross these 2 organisms together? What do their offspring look like? And you don't want to have to wait, months and months and months to get the answer, right? If you can wait a week instead or a few days, that's ideal. And fruit flies are tiny, so you can have tons of them in one lab. So they have been very well studied. So I think the simple way to put it. So I'm not going to dig into all the nitty-gritty of what Thomas Hunt Morgan did, but I do want you to at least get kind of the big picture because it is a huge deal in terms of biology and genetics. And so in a nutshell, what Morgan and his students were doing is they were doing dihybrid crosses with fruit flies. And remember, dihybrid crosses is looking at 2 different traits, 2 different genes at the same time. So for example, in this instance, they were looking at the body color of the flies, black versus gray, and then kind of the shape of the wings. So quote unquote normal wild type versus what they call vestigial, sort of sad shriveled up wings. And essentially what they found doing these dihybrid crosses is they were not getting the expected Mendelian ratios. They were getting weird exceptions to that. They found, you know, versions of these two traits seem to always tend to kind of stick together. So like the gray body color was almost always associated with normal wings, and the black body color tended to be associated with the vestigial wings. And that's not what you would predict through Mendelian crosses. You're not getting those 9-3 to 3-1 dihybrid ratios that Mendel got. And so the brilliance of Mendel and Thomas Hunt Morgan and his group is they realized that, well, what could explain these exceptions is if the genes controlling these two traits that we're looking at are actually physically linked together, located on the same chromosome close to each other. Something like this. So in Mendel's example, if he was looking at two genes, right, doing a dihybrid cross, the S gene and the Y gene, we just assumed that those genes were located on separate chromosomes. You might remember Mendel's law of independent assortment, right? The two homologous chromosomes pair up that have the two versions of the S gene. The homologous chromosomes with the two versions of the Y gene pair up, and they could orient themselves in one of these two ways. But the bottom line is, this pair of chromosomes is not paying any attention to what this pair is doing. The S gene is assorting independently of the Y gene. That was critical for that F2 9-3 to 3-1 ratio that Mendel always got. But like I said, that's not what Thomas Hunt Morgan and his group were getting, and they realized, well, what if instead of looking like what we just saw in the last slide, it was something like this, where the body color gene and the wing shape gene were actually linked together on the same chromosome. So we're just dealing with one chromosome pair here, and if that's the case, they're not going to assort independently from each other. They will tend to be inherited together is the bottom line. So that was a big deal because not only was that an important exception to Mendel's rules, but it was also really led to this understanding that, genes, these factors that control these phenotypes are actually physically located on chromosomes. Before this work, nobody even really understood that. So it was a big breakthrough in genetics and in biology in general. And his group took it even a step further, and they realized, well, even though these two traits do tend to be inherited together, sometimes it looks like they are sort of assorting independently and getting separated. And they realized, well, okay, if these genes are located on chromosomes, this could be due to these crossover events between the two genes. So we end up with these little recombinant chromatids, right, where we do occasionally separate the two recessives and the two dominants from each other and get these kind of heterozygous sperm or eggs being formed. Okay, and they realized, okay, they called that recombination, that crossing over between the two genes. They realized that the frequency with which that recombination happens was basically directly correlated with how close or how far apart the two genes were from each other on the chromosome. So they were calculating these recombination frequencies, and they understood, these frequencies are actually telling us how close or how far apart the two genes are. And that kind of makes sense. Let me go back here. Imagine these two genes, the B and the VG, are like very, very closely linked, even closer than they are in this picture. If that's the case, what's the chance that you're going to get this crossover between the two genes? It's going to be very small, right? Because there's just very little space there to work with. Whereas if these two genes are on the same chromosome but they're very far apart, it's more likely you're just going to get a crossover between them just due to random chance. So that's kind of the basis for those frequencies. So higher frequency, genes were farther apart. Lower frequency, genes were closer together. And Morgan's group realized, well, we can actually use this to actually start mapping out how far apart genes are from each other in the fruit fly. So they just started doing tons of these dihybrid crosses, looking at different gene pairs, and they started building these rudimentary maps. And this was the first time this had ever been done. So the distance between two genes was referred to as map units, or sometimes it got called centimorgans instead of centimeters, right, for Thomas Hunt Morgan. Clever little pun there. But anyway, this was huge. This just blew up the world of genetics. So it's like, oh, we can start mapping these genes out and figuring out where things are on the chromosomes. Of course, this kind of technique is totally outdated now because we can just sequence the whole genome and see where all the genes are. But it was a big deal at the time. All right, let me stop there, and I will finish this up in the last video.